All right, so we were talking about experimental error. The treatment structure. So here you can look at a simple treatment structure. I've got 4 plants, all of them getting the same amount of water, the same amount of light, the same pots. The only thing that varies is the fertilizer. Oops. I have fertilizer represented by this drop, this green drop. So I've got no fertilizer added, 2 drops, 4 drops, 6 drops. So in this example, my independent variable, the thing I'm changing is the level of fertilizer per plant. My constants are the water, the pot size, the soil in the pots, I'm sure, and the light that they are receiving. And also, I'm sure, the genetics of those 4 plants. I'm going to try to pick 4 seeds that are genetically as identical as I can get. So remember, the independent variable is what you change. What did the scientist change? The dependent variable is what did I measure or observe? And the constant is what did I try to keep the same? And I think this previous slide does a good job of that. I've got the same pot size, same soil type, same plant genetics, same light source, same water. I'm only changing fertilizer. Fertilizer is my independent variable. And what am I measuring here? You can't see my dependent variables. Maybe I'm measuring plant dry mass. Maybe I'm going to measure yield of beans off of these plants. I don't know. Maybe I'm going to measure color. And there's a lot of things I could measure. And then constants, light, water, pot size, genetics, soil. All right. So those are the types of variables that go into an experiment. The control group has no independent variable from a strict statistical standpoint, experimental design standpoint. You could say that's true, but the independent variable could be held at what we call normal. In agriculture, that happens a lot, where our control group is whatever we normally would do, our normal fertilizer routine, our normal pesticide routine, whatever we've been doing is considered our control group. So is it getting an independent variable? Technically, you could say, well, yeah, it is. If we're measuring fertilizer, is it getting any fertilizer? Of course it is. But it's not getting a changed or a different independent variable. So no change in the independent variable. It kind of depends on the experiment and which industry you're talking about. The language of experimental design is a little different in the medical community versus the ag community versus the, what is it called? Political science or social science communities. They're very subtly different, even though lots of things are the same. But we're still going to measure the dependent variable. I don't care if you've got the control group or the test group, you're still measuring the dependent variable and you're still having constant variables. The control, the only difference is you're not changing that independent variable. So we're not going to go through a practice example here. This would be normally a time to have a discussion in class, maybe breakout work groups or something like that. We're not going to do that. So basic principles of experimental design. First, you need a valid question. First, you have to observe something and come up with a hypothesis and then make a prediction. And that prediction is your valid question. What do you predict? What do you think is going to happen if you run some hypothetical experiment? And if you get through the mental process of a hypothetical experiment, then you can start designing it and make it real. You need to have your control or comparison group. You need to have replication. So for example, if I have one plant that gets no fertilizer and one plant with fertilizer, and the plant with fertilizer dies, do I say fertilizer is a bad thing? If that's all the evidence I have, yeah, I might come to that conclusion. but that's not good enough. You can't actually just look at one plant in the control, one plant in your treatments. You need to replicate. You need to have multiple observations. So if anything goes wrong with one group, or the background noise, the constant variable noise that you can't control, might confuse you. You can't see that without replication. So you always look at things multiple times. And frequently, I think 3 or 4 times in a single experiment is the minimum. And you probably want to do the same experiment at least twice. So if I'm running an experiment, for example, in lettuce, and I say that lettuce a lot because that's where a lot of my experience is, so it comes to mind, I might run the same experiment where I look at it 4 times in one field in 4 different fields. So 4 times 4, I actually looked at the experimental treatments 4 times 4 or 16 times before I'm done with that year's look at it or that season's look at it. 4 observations in a single experiment, and then I want to look at that same experiment another 3 or 4 times. That's replication. Now, those are just generic numbers. How much replication is really necessary? That's where you need to know what you're doing. You need to have some experience. You need to have a statistician on your team, because that number, 4 replications in 4 different trials, that's just a number I came up with that I felt comfortable with in certain types of lettuce trials I did. Other experiments are very different. You might need much more or less in order to come up with a valid answer to your question or experiment. Randomization, that's also very important. If I'm giving sugar pills or medication in a double-blind medical study, you can't pick the people on your own. You have to randomly assign half the group to one side and half the group to the other without any accidental bias by the experimenters. And the same thing is true in the field. I randomize things. So if I'm going to do a trial out in the field, I might draw a trial on the paper, and I've got a grid. Let's say there's 16 plots in my trial, and I got 4 treatments that I'm going to look at 4 times, 16 blocks of plants in my lettuce field. And I'm going to be doing some sort of a spray to hopefully control a root fungus. Well, which ones are going to get the spray and which ones aren't? I'm going to randomize that either using a tool like Excel's randomization function, or what I used to do in the old days, I had 120 pennies that were labeled with Sharpie 1 through 120. And I would just say, all right, how many do I need? And I would just shake them in a bag and reach in and grab. All right, the next treatment goes to block number 3. Next treatment goes to block number 47. And I would just pull the pennies. And it was just a fun exercise, sit back after a hard day's work with a group, and we're going to randomize the next experimental design, lay it out, and you can sit back, chill, randomize with a beer or something, and it's easy. But now with some of the tools we have today that are so easy, I haven't used the pennies in a long time, 25 years. I still have the pennies, but I haven't used them because Excel is so easy to do randomization. And then we need stratification or blocking. And I'm not going to get into that at the moment because blocking is its own discussion that I'll get into in later slides. So, examples of a question. The question, does magnesium affect shoot length in bean plants? So the shoot is the above ground portion, roots below ground, shoots above ground. Does magnesium affect the shoot length in bean plants? So I select a bean variety in a stage of development that I want to measure. So do I want freshly germinating seedlings, or do I want to run the experiment on a little bit older plants? It's up to the experimenter. What do you think based on your knowledge? Define the experimental unit. Am I going to look at groups of 5 plants, 20 plants, 1 plant? What is my experimental unit? I'm going to determine my statistical analysis, and we won't even get into that in this lecture, but you need to know what are the statistics you're going to run before you even start the experiment. And I've had this experience a number of times where somebody comes to me and says, we've run this experiment, we have this data, and we're not really sure how we can properly analyze it. We're a little stuck. We're not getting the results we want. And it's heartbreaking for me to look at somebody's work, which might have hundreds of hours of labor and all the cost and all this data they've collected. And the answer is start over. Throw this out and start over. Let's make a good plan, because you can't turn garbage into useful information. Now, sometimes there's a way to get something out of it, but I never want to give somebody false hope that there is useful data. And I really don't want to run statistics on a bad experiment, because statistics will always come up with an answer, always. The problem is, if the data isn't sufficiently complete or wasn't set up right, the statistics are giving you a bogus answer. And I never want to tell somebody, all right, let's run the statistics on that. The statistics say, you know, fertilizer A is much better than fertilizer B with a 87% confidence rate, when I know the reality is the data might say that, but the data is garbage. The reality is we have no clue. Start over. So a good design, you always know what your design is and what statistics you're going to run before you even start anything. So I've done all of that. I know what I'm going to do. So I provide the bean plants with my added magnesium. I've got a little kick with some extra magnesium in the non-control units. Don't forget, I got control units. I wait my 14 days. Again, where did that come from? That's part of the experimental design and knowledge about bean plants. And then I go and measure the shoot length and I gather my data. So the comparison control, compare the bean plants given 1% magnesium to bean plants given only water. There's my control, my comparison. Compare garbanzo bean plants, chickpeas, with 1% magnesium to black bean plants given 1% magnesium. I could do that. And if I wanted to get a little fancier, I could do garbanzos and black beans with 1% and garbanzos with water, black beans with water. So now I got 4 different things to look at, a little more complicated. But essentially, you're running the experimental group concurrently or at the same time in the same experiment. You're running the comparison. I don't say, well, you know, last month I grew a bunch of garbanzo beans and measured them without this magnesium, so I'm going to grow a new set of beans with magnesium, and I'm going to compare the beans I grew this week with magnesium to the beans I grew last month that I had no magnesium. No. You have your controls at the same time. Same experiment, same conditions, same everything. No matter how good you think you are at keeping constant variables, making sure that the soil's the same, the weather's the same. Can you control the weather? Oops, no, you can't. Well, I can in the greenhouse, sort of. You can't do it perfectly. So always run your controls right next to at the same time as your treatments. The word control is used in 2 ways. So I have this additional slide here because we do use the word control in a very casual way. We want to control experimental error. We want to control that constant variables to keep them perfect. And I don't like using the word like that too often, but I I just do it out of habit sometimes, and a lot of people do. The constant variable, you want to keep it constant. You want to keep your soil type exactly the same. The amount of water each plant gets exactly the same. The seed planting depth exactly the same. The temperature, the sunlight, you want to keep everything the same. You want to control all of those things that can lead to experimental error. And we'll talk about experimental error in a moment, but experimental error isn't a mistake. It's just the background noise that you can't help. Remember those 100 lettuce plants I talked about earlier? If I grew them exactly the same, are they going to end up being exactly the same size? With weight, with exactly the same core height, with exactly the same color, with exactly the same shape, exactly the same number of leaves on each head? No, there's going to be some variation in those plants, some noise, some experimental error. Now, I'm not going to test you on this, but for those of you really trying to wrap your heads around this, the constant variables are those factors I'm trying to keep the same: the soil, the moisture, the sunlight, the temperature. I'm trying to keep those the same. The experimental error is the noise in my measurements, in my dependent variables. They're closely related. The control, excuse me, not control, sorry, the constant variables are never perfect, and so you get changes in the dependent variables. Those unavoidable changes in the dependent variables are experimental error. So this number one definition on this page, controlling that experimental variable, or controlling your constant variables, is, I think, a poor use of the word control, because in statistical design and statistics in general, the use of control should really be used as the experimental control, the control group, the control receiving no independent variable or no change to, quote, normal for the independent variable. Are you confused yet? This is tough stuff, I get it. I'm going to be gentle on questions I might ask on a test on the final exam, but you do need to try to wrap your heads around this, and it might take some writing things down, going through the PowerPoint more than once. Pay attention. So before I get to examples of controls, I'm going to take another break.